In this video, we're going to talk about the complement system. The complement system is made up of a series of proteins, and these proteins are produced in the liver, and after they're produced in the liver, then they enter the bloodstream and they just float around looking for pathogens. And there are a lot of proteins involved. This is going to be a very basic overview. So the main proteins are termed C1 through C9, and the C stands for complement. So complement protein C1 through C9. And when they're first produced and they first start circulating in the body, they are in an inactive form. So that's why they're just constantly moving around in an inactive form, and they do not activate until they encounter a pathogen. So they will activate in the presence of a pathogen. And again, these proteins, C1 through C9, they were named in the order they were discovered, not in the order that they work. Okay, so here we go. So again, here's the liver. It's producing these proteins, C1 through C9. They get into the bloodstream. Here's the bloodstream. And these proteins are just kind of floating around, looking out for any pathogens. Complement is part of the innate immune system. So they are not specific. They are just constantly searching for any pathogen that gets in. Okay, you're just hanging out. Here's your skin. Don't forget your skin is very thick, many layers of cells. Okay, and you come into an accident and here comes the pathogen. All right, you have a tear in your skin somehow and the pathogen enters. Okay, so the pathogen's going to enter and it maybe gets into the blood. Now, the first thing that's going to happen in the complement system is it needs to get activated somehow. And there's three ways the complement system gets activated, three pathways of activation. The first pathway is called the alternative pathway. The second pathway is called the lectin pathway, and the third pathway is the classical pathway. And I'm going to explain how each one of these pathways gets activated. Okay, so here's the pathogen. Let's pretend like this is a bacteria. All right, so here's a bacterial cell. Now there's a lot of antigens on the bacterial cell. Okay, so these are different antigens. The bacterial cell also contains carbohydrates on the surface of the cell, and one of these carbohydrates is called mannose, and mannose is a unique sugar that's found in the bacterial cell wall. So let's talk about each of these pathways. Let's start over here with the classical pathway. Classical pathway gets activated when an antibody binds to an antigen on the cell surface. So this gets activated when an antibody binds to the antigen. All right, so when this happens, this causes the formation of an enzyme called C3 convertase. C3 convertase forms whenever antibody binds to an antigen on a bacterial cell. So that's the classical pathway. The next pathway let's talk about is the lectin pathway. In the lectin pathway, a protein in a human called a MBL. This stands for a mannose binding lectin. Okay, so here's the MBL, we'll put it in red. This protein is in the human body, and it's a big protein, and it's going to bind to the mannose on the bacterial cell wall. So it binds to mannose on bacteria. And again, when this happens, C3 convertase is formed. The last pathway, the alternative pathway. This happens all the time in your bloodstream. There's a little bit of C3b, complement protein 3 subunit B, floating around. As C3b is going to stick to the pathogen randomly. When C3b binds to the pathogen, this activates the alternative pathway. And again, just like in the other two, when C3b binds to a pathogen, C3 convertase is formed. Okay, so all three of these pathways, they do different things, but the end result is C3 convertase is formed. So now let's talk about what C3 convertase does. The complement system is a cascade of events. The first protein involved is C3. When C3 convertase is produced, it's going to split C3 into C3A, complement protein 3 subunit A, and C3b. Okay, so C3 convertase causes this split to happen. That's the first step in the cascade. The next step involves C5. Once the complement system recognizes that C3 has been split, C5 convertase gets produced next. C5 convertase is going to split C5 into C5a and C5b. Now that these proteins have been split and produced, the complement system can start acting and fighting against the pathogen. So a few things happen after activation. So results, outcomes of the complement system activation. The first outcome is that the inflammatory response is going to be enhanced. So inflammation enhanced. And there's two proteins in the complement system that enhance inflammation, and they are the C3A and C5A. So C3A and C5A are going to enhance the inflammatory response. And here's what happens. C3A and C5A are going to cause the blood vessels to dilate. Okay, so they enhance the permeability of blood vessels. And the second thing they're going to do is they're going to attract phagocytes to the site of the pathogen. So when C3A and C5A are produced, the blood vessels increase, become dilated, and macrophages, okay, here's a big macrophage, and neutrophils can move to the site of the pathogen. So here's the pathogen, here's a macrophage, here's a neutrophil. C3A and C5A allow them to move faster and toward the site of the pathogen. So that's the first outcome of the complement system. The second outcome is called opsonization. Opsonization is a fancy word that just means easier for phagocytes to engulf. The part of the complement system that enhances opsonization is C3b. Okay, so once again, here's our pathogen. Once the complement system's activated, a lot of C3b is going to be produced, and the C3b is going to coat the outside of the pathogen. And the more C3b that's produced and attaches to the pathogen, the more C3 convertase is formed, okay, and again, more C3b is made. Now that C3b has coated the pathogen, here comes a macrophage. Macrophages have special receptors that will nice and easily bind to the C3b, and then they can gulp up the pathogen and eat it and chop it up and kill it. So opsonization is the second outcome of the complement system. The last outcome of the complement system is called a membrane attack complex, or a MAC. MAC involves the rest of the complement protein, C5b, C6, C7, C8, and C9. And again, it's a cascade. So once C5b comes in, then C6, then C7, then C8, then C9. Here's what a MAC does. Here's our pathogen. What's going to happen is C5b is going to come, then C6, then C7, then C8, and then a few C9s, and they're going to essentially punch this hole in the bacterial cell, and now what's going to happen is the cell is going to lyse. Okay, it causes cell lysis. And again, obviously this will kill the bacteria, kill the pathogen. All right, so I hope that helped in explaining the complement system.